right, awesome. Well, we will just wait 1 minute. Welcome everybody until we get everybody in here and loaded up. I think we're good to go. John, can you hit the next slide for me? Awesome. All right. Well, welcome everybody to today's presentation on DNA fingerprinting through variable nucleotide tandem repeat technology, or for short, VNTR technology. Thanks for everybody for taking the time out of your day to come to Segra's second webinar. My name is Carson Otto. I'm a director of sales here at Segra, and I'll be co-hosting today's webinar with Mitch Gallen. Right now, we're sitting at about 30 attendees, and judging by the registration list, it's a wide range of individuals across the cannabis industry. Now, for those of you who are unfamiliar with Segra, we are a tissue culture nursery with two core divisions, a tissue culture division located in New Westminster, BC, and a molecular lab services division located in Richmond, BC. Now, John, could you hit the next slide for me? So, as for the technology, the genotyping technology that we'll be discussing today, we routinely perform this operation at our Richmond facility, and it is an integral process while producing tissue culture plantlets and validating genetic identity prior to shipping them to our clients. Now, for example, if we were to accept plants from a client under the Regenerate and Preserve program for initiation and cleaning in tissue culture, we would use this technology to establish a reference sample and take a DNA fingerprint. Now, with this DNA fingerprint, we create what you can see here on this slide, the Segra Certificate of Genetic Identity. So through the DNA fingerprint and during the initiation process, we can protect valuable IP, of course, but as well as monitor for somoclonal variation throughout the tissue culture multiplication. And before sending it back, and once the genetic is clean, we can validate the genetic once again and make sure it is what our partners sent us. Now, outside of internal QA purposes, we also offer this service as a low-cost, fast way for breeders and cultivators to both track plants in their facility and also validate cultivar identity. This is done by simply sending us a reference sample. It's the size of a peppercorn. And then you too could also get one of these Segra Certificate of Genetic Identities. So with that, I'll hand it over to Mitch, and he'll give you the rest of the details on today's presentation and what you can expect for the next 57 minutes of this webinar.
Awesome. Thanks so much, Carson. Today, our Chief Science Officer, Dr. John Brunstein, will be running through a presentation focused on the background, process, and application of our VNTR DNA fingerprinting test. In addition to developing and validating this test, Dr. Brunstein runs the molecular lab division and facilities here at Segra. Before joining the team, John both earned his PhD and taught at UBC as a professor in the Department of Pathology and served as CSO at multiple biotech companies. He's considered a leading expert in the field of biotech, molecular biology, and clinically related topics with an extensive publication history. Following his presentation, there will be a 20 to 30 minute Q&A period in which we'll answer audience questions. Feel free to type any questions into the box titled Q&A at the bottom of your screen as they come up, and we should be able to get through all of them in the end. If you want to learn more about this topic or any of our other services after the presentation, Carson and I would be thrilled to connect directly in chat, so don't hesitate to get in touch. And without further ado, it's my pleasure to introduce you all to Dr. John Brunstein.
Okay. Well, thank you, Mitch and Carson, for that introduction. So you sort of already heard this, but a quick background as to where I come from in this field. My undergraduate degree was at Simon Fraser in biochemistry. My doctorate was at UBC in biochemistry, where I specialized in molecular biology of viruses. I did a postdoc at the University of Helsinki in clinical molecular virology, and then I was UBC faculty for 10 years in both biochemistry for a while and then pathology and lab medicine. Concurrent with some of that, I was chief scientific officer at the Provincial Health Authority's Center for Translational and Applied Genomics, where I was doing a lot of molecular lab development and deployal. In 2010 onward, I ran my own company, which I actually still have, which is consulting for private industry molecular diagnostics in clinical work, and also compliance and laboratory quality systems. And all of that was kind of a good basis on which to build. So back in 2015, I joined Segra. I think I was about the fourth person to join the company. And the idea was to start taking molecular biology and some DNA fingerprinting techniques and the ideas of quality systems and apply them in the cannabis space, which was kind of a free-for-all then, and try to get a little bit more uniformity and objectivity in terms of variety definition and sample tracking and quality assurance. Okay, so with that out of the way, we're going to cover only a handful of key topics today. The first of those is, why is it useful to have an objective means to identify specific cannabis clones regardless of their growth stage? So whether it's a little tiny sprout that looks like an alfalfa sprout, or it's a full-size plant, can you actually identify that as a particular clone? Yes, we'll show you why. And why would you want to do that? Okay, well, the next thing would be, what is a DNA fingerprint and how does it provide an answer to the why above? How does a VNTR method for getting a DNA fingerprint differ from other methods? And there are several other methods I'll touch on briefly. And in particular, how does it differ from the end user perspective? I don't think any of you are probably really interested in the technical differences. It's how does it matter to you in the way you're going to apply this, and what does it give away or not give away that you maybe want to have some control over? And of course, if you're going to use a technology, you shouldn't just know what it does. It's also very important to know what it doesn't do. So finally, what doesn't a VNTR result tell you, and how is that useful to put into your equation? I'd like to start off with a real life scenario here. And sorry, I don't know if everybody can see that video, but it's blocking the text I have to read here. The real life scenario here, this is something that we've actually dealt with. A cannabis cultivator, I won't say who it is, but they approached us. They were using traditional mother and cuttings propagation, and they had a particular high value proprietary clone that, like many cultivators out there, they were very proud of this. They really liked it. This was their main product. And at this point, they had 55 mother plants, but they were concerned about long-term product uniformity. The question being, were all 55 plants truly clonal, or were some of them non-isogenic? So as an aside, the question you might want to ask yourself is, how would this happen if they're all cuttings from mother or cuttings from a cutting of a mother? How do you get this? You get it through point mutations at, let's say, near a node. You take that cutting, there's now a number of cells generate out of that mutated cell. And so now you have essentially variegation within the plant, and depending on where you do the cut and where that mutation was, you can get cuttings that are no longer really true to form. And a hint, this is not called genetic drift. When we get to near the end of this, we'll give you some more information on where you can look up why this is not genetic drift and that term is misused. But the bottom line is, in a case like this, how can the cultivator do a quality assurance test to verify that all these mothers are true to type? And if they do it, what do they find? Another real life scenario. This is a different cannabis cultivator, again using traditional mother and cuttings propagations, and they had multiple clones. Let's call them A, B, C, and D. And these were due for an upcoming production run and at different scales. So they wanted so many A, so many B, so many C, so many D. And they set their crew in to make cuttings of the mothers. And I don't know how, I didn't bother asking, but somehow these cuttings all got just scrambled up. It's like they're in a bucket and they're a bunch of cuttings. They're visually indistinguishable. So which one is which? So how does a cultivator untangle this mess and get the varieties separated out so that each one gets the right production, as opposed to having them all mixed up on the production floor? Okay, I'm going to come back to those, but before I do, I'm going to have to talk a little bit about how we would address those questions. Let's start by thinking about some basic biology here. DNA is effectively the genetic repository of instructions for a living organism's cells. It's the blueprint, if you will. And meaningful variations in DNA, there's a lot of variations that are not necessarily meaningful. They're in non-coding regions, non-informative DNA, but meaningful variations in DNA between two organisms, such as two cannabis varieties, can be broken into a number of types. These can be in unique genes that are unique to one plant. So one plant has that gene, the other one doesn't. They can be in a shared gene. So both plants have the same gene, but they can have different forms, or what we call different alleles of that gene. Or they could be in regulatory elements, which are not strictly genes themselves, but they're DNA fragments or sections that control the expression of other genes. They turn them on or off or control their levels. These particular combination of changes essentially constitute the variety genotype. And interactions of the genotype with the environment leads to the observable parameters such as size, color, growth, behavior, and chemotype, which we describe, these observable parameters are what we describe as the phenotype. Well, newsflash here, guess what? Your end consumer really just cares about the chemotype, right? They don't really care the product they're getting. They want to know what's in it, what are they going to get, is it reliable, is it the same thing? The master grower has a somewhat different concern. They actually care about the phenotype. They could have two different varieties with the same chemotype, but those could have very different aspects of disease resistance or growth cycle or yield. One might be much better than the other for growth and production. So the master grower really cares about the phenotype in order to get to this final chemotype. And in order to reach a desired uniform chemotype, the master grower has to achieve target phenotype through the environment, the nurture, part of the what's usually called nature and nurture. But they have to, this controlling environment has to be on a stable, uniform genotype so they know what the outcome is. So they can control the environment, they can't control the genotype, so they had better start with a uniform, correct, known genotype. Those together give you a uniform phenotype, and then your end product, your chemotype, stays unique, and you have brand regularity, something that's, lot to lot, going to be reproducible, which is what you're trying to get. Okay, so this leads us into DNA fingerprints. Well, plants don't have fingers, right? Okay. Well, we use the term DNA fingerprint by analogy. And cannabis has a diploid genome of around 1.8 gigabases. That's too big to sequence the whole thing. It takes a lot of time. There's a lot of cost. There's a lot of bioinformatics requirements. And frankly, most of it is non-informative and not very useful. If you'd like to be able to use DNA as a way to identify individuals, what you need is a unique DNA-based biometric identifier. And this is analogous to a fingerprint in that it needs to be simple, fast, cheap, reproducible is obviously a real key here, and it needs to be unique to an individual clone. One of the things that's important to stress is that a fingerprint doesn't mean anything. It just identifies a person or a plant. And, that's where the fingerprint analogy is very nice because, as I say here, a fingerprint doesn't tell you the person's eye color or hair color. It doesn't tell you these other phenotypic traits, but it identifies the person. And that's going to be useful because maybe you don't want to give away your phenotypic traits. There are a lot of different ways of doing DNA fingerprinting, and sort of roughly from cheapest and simplest to most expensive. I've ranked them here on the left, and then I've given a chart where I've described their cost, their complexity, their power of identification, and their ability to disclose linked traits. I'm going to talk more about this linked traits issue in a moment. So at the bottom of the list, we have methods like rapid and ISSR. These are very cheap, but they're also very low throughput and they're poor reproducibility. There have been published papers using these in cannabis. We played around with a little bit here in the very early days in the lab, and yeah, they work, but they're not suitable for industrial scale and reproducibility. VNTR, the method that we're going to be focusing on today, is still quite cheap to do. It has moderate throughput. It has excellent reproducibility, and it does not yield linked traits. I'm going to hammer back on what that means in a bit more. Another method that is used in this space that we don't use is single nucleotide polymorphisms, or SNPs. This is a little more expensive than VNTR. Setup is actually a lot more expensive. So moderate cost, it's high throughput, but it's very inflexible, and it does yield linked traits. The next sort of level up is GBS, or genotyping by sequencing, which is high cost, low throughput, and again, yields linked traits. And at the very far end of the spectrum, you have whole genome sequencing, WGS. This is the highest cost, the lowest throughput. And by definition, it's not just linked traits, it's data on all the genes. It's the master copy of everything if you know how to interpret it. So, I've talked about linked traits. Let me reiterate what those are. So this is akin to the fingerprint telling you eye and hair color. Cultivators, like in our two cases I started with here, don't necessarily want to give away a bunch of information about their clones. They would like to know identity, but they don't want me to know that their key thing is high myrcene, high CBD, it's fusarium resistant, all these things that link traits might tell me. In particular, if they're going to share information, some sort of identifier with someone else, they don't want to be giving this away. VNTR provides a sweet spot. Not only is it cheap and extremely reproducible with very good resolving power, but it has essentially no capacity to reveal link traits, whereas SNP and GBS don't provide any benefit in identity resolution. They're mathematically somewhat better, but their VNTR is already far past what's a meaningful resolving power, and we'll talk a little bit about that. So you get unused additional mathematical on paper resolving power in return for giving away a bunch of information that you might want to keep closely held. So summary, why VNTR? It's arguably the best combination of resolving power, cost effectiveness, reliability, reproducibility, and flexibility, and this lack of disclosure of linked traits. But hey, you don't have to take just my word for it. Earlier this year, a thesis was published by a Dr. I think it's Rachel Houston. She spent 4 or 5 years doing a thesis purely on genetic identification of cannabis varieties. And when I looked through that, I found the following quote, which I have here in green. Capillary electrophoresis, CE, of STR markers is the gold standard for the genetic identification of marijuana. Okay, well, yes, there are far too many acronyms in science, and her phrase there, CE of STR, is simply another acronym for the VNTR approach. So this is accepted, and there's a number of other papers out there with similar comments. This is accepted industry-wide among knowledgeable molecular biologists and geneticists as really the best way to handle cannabis identification. So how does it work? Well, I'm not going to go into the molecular details, but VNTR stands for variable nucleotide tandem repeats. And what it does is it measures the size of a small number of highly variable repetitive elements in a sample. Cannabis is a diploid organism. There's a maternal and a paternal copy of each chromosome. So 2 chromosomes, 2 copies of each one. And so it's possible to have 2 different versions of a gene that's on a particular chromosome. You can have a maternal version and a paternal version. So we can get heterozygosity, or 2 different sizes of this. Or you could have them both be the same size, which is homozygous. If we look over here, this is an example of 3 different cannabis varieties and a single marker. The orange peaks here are an internal size standard that's injected, and that's how we can tell we're looking at the same size range here. The blue peaks are a particular marker out of 3 different varieties. And what you can see is that in this particular clone up here, these 2 markers are different between the maternal and maternal sizes. This has a 90 base pair and 101 base pair versions of that particular marker. This one has a 90 and a 96. This has 2 copies of 90. And for any geneticist in the audience, yes, it could be hemizygous. It's possible it's a 90 over a null, although you'll note the peak height here is suggestive that this is a true homozygous 90-90. The bottom line is you don't have to be an expert at this to look at the first one, the second one, the third one, and say that those are not the same. So it's easy to identify differences. Well, we don't just use one marker. We actually multiplex this across multiple loci. So here's an example of a trace showing one marker in blue, one in red, one in black, one in green. The black and the green one are both homozygous in this particular clone. This is the red's heterozygous, the blue is heterozygous. And we do this across, in the current version, 12 different markers. And this gives us probability of identities ranging theoretically from a 1 in 14 million chance of a random match to a 1 in 10 to the 49 chance of a random match. And those are theoretical bounds. Actual samples run at about a 1 in 100 million chance that that particular string of numbers would come out of something that's not actually the same clone. So far more than is need to actually be 100% positive that you have the right thing. And nobody's going to see the actual numbers in this chart here, but what you can see is you get the idea that we have a particular clone and what we call a C number, which is a cannabis DNA sample number. And then there's this whole long ordered string of values for all these things. We call this a VNTR vector, and that is kind of the identifier of it, of that clone. So what does one of these tell you? Well, it tells you relatedness between samples. Tells you if 2 things are clonal or isogenic, they'll get this same string of numbers. It can also tell you if something's a near neighbor. They will have that string of numbers will be different, but there'll be a very small number of differences there. versus something that's a distant relative where a lot of those numbers are going to differ between the two of them. So how do you visualize these? Well, what I'm showing here on the right is something called a phylogenetic tree. Phylogenetic trees are easy to misunderstand, but the basic concept is pretty simple. Closely linked samples, and by closely linked, they'll have a very, very short link between two of them. And again, you can't see details, but there are two things right there. Closely linked samples are genetically very similar and will have generally similar traits. Things that are identical, like things are all along one line here, these are all the same clone. All of these over here are the same clone. There's like, last count, like 19 different names we've seen applied to this clone, but they're all the same thing. Okay. When two things are not identical, they will have some differences, and we can't say from this ahead of time what those differences are. There's a lot of different traits that make up a phenotype. If things are very close genetically, most of those traits will be really similar, but a few won't be. And we can't say ahead of time if the traits that are different are ones that are important to you or not. So you have to assume there may be some differences. Maybe they're important, maybe they're not. But in general, most traits will be similar. The further apart you get genetically, the less and less similar two things are. Okay. Another way you can visualize this, and it was, I won't say who the company was, but there was another company in this space that used something called principal component analysis, which is very nice mathematically, but what it does, it takes your data and it creates a multi-dimensional space out of it. When I've done it with our data, it wants to go to a 72-dimensional space to fully describe our data set. And then what you do is you take your most important, most informative dimensions out of that. And the answer is, how do you try to display things on a two-dimensional screen, like a computer screen? And so you can do left, right, and up, down, your X and Y axes, okay. And on a computer screen, you can kind of zoom in and out of it and kind of in a three-dimensional space. That's now a third axis. And you could use a color spectrum as a fourth axis. And so they did that. And it was kind of this three-dimensional space you could float through with things of color. The problem with principal component analysis, it looks really cool, but it's kind of Fisher Price. It's something you put in front of a baby because it looks cool, but it doesn't do much. Here's the reason. If you do principal component analysis on this data set and you take those first four axes, they only account for about 30% of the genetic variability, meaning that any two spots that are coincident in X, Y, and Z and the same color, and you look at them and think, hey, wow, these are the same thing, those can actually be more different. They can be 70% different and only 30% similar. So it can be kind of misleading. So we normally use phylogenetic trees. Okay, now we understand a little bit what we can do here. Let's talk about the real life applications. So in scenario one, remember, this was somebody had 55 mother clones. Are they all the same? 54 of the 55 were isogenic. And actually, as an aside, we usually encourage customers, or I certainly go out of my way to encourage customers, slip a few known surprises in there just to double check that what we're telling them is real. And they actually sent us 56 samples, and one they knew was an outlier, and they were pleased when we said, hey, one thing is completely different. And they're like, oh yeah, we knew that, we were checking it. Anyways, but of the 55 that they were actually interested in, 54 were isogenic, but one wasn't. It had these very small but observable genetic changes from its fellow cuttings. And, kudos to this producer. They immediately culled that one. It was a loss of, less than 2% of their mother stock, but it ensured that the rest of the material being grown was uniform. So this helps maintain brand and product identity. Scenario two was the bucket that the junior cutting guy had presumably scrambled everything into. And so what the cultivator there did is we sent them sample kits, and they went to the known mothers and sent us what would be reference samples of each of those. And then they followed up with pieces of each of these cuttings. And as it was alluded to in the beginning, we have these sample kits, and it's literally about half a peppercorn's worth of material you need to send us. And so what we do is we use the reference samples. We did the VNTR. We got a VNTR vector off each of these. And then we tested each of the cuttings. And within a couple of days, we were able to unequivocally identify each cutting back to its source. They were able to properly reassort the cuttings and get things back into the right production cycle. And again, meet production goals of specific and uniform product. Again, sort of the key here is helping maintain brand and product identity. Okay, well, what about other VNTR applications? Well, as was also alluded to in the opening, we use this for variety tracking and quality assurance. So any incoming material here for whatever program, I like to get a sample of that as soon as it comes to the door. Get me a little tiny piece of that, and we can now lock that down. We're not really talking about it here, but we take that long string of numbers that's hard to read, it's clumsy, and we have a way to generate something we call a variety identity code, which is a 32-character string derived from that. It's kind of like a serial number, and it's easy to look at, and it's easy to see if it's the same or different between two things. And so we get this data, we get a variety identity code, it goes on a genetic identity certificate, and now we can track it in production. In particular, when you come back, because you're now ordering stuff either back out of the regenerate and preserve, or if we're doing large scale production and you've ordered a lot, these little plants going out, we can test and we can objectively say, yes, this is your clone. And this is really important because little plantlets of ditchweed industrial rope hemp and psychedelic flaming space monkeys number 7, which I don't think are real variety names we have, but they sound like good variety names to me. If I had varieties, that's what I'd call them. But the point is they'll look the same. And if you're the producer, you need to know that you're getting your clone and that you're devoting these resources 4 months down the line. You don't want to find that you had the industrial rope hemp when it was supposed to be flaming space monkeys. Okay. So that's internal. Externally, the sort of things we tend to do more on the service side is for variety confirmation with cultivators. And one of the things we can do is if you're a cultivator and you've just paid a whole bunch of money for a clone, did you get the right thing? And if you can get us samples of the parental stock and what you were sent, we can tell you if it's isogenic, maybe you expect it to be an exact clone. We can also tell you if it's really close, and maybe you got seed and you understand it's going to be close, but how far apart? And we can tell you, yeah, this is close. Or no, you know what? You got floor scrapings. This is completely not the right thing. We can do variety identification to a voucher specimen. So if somebody gives us something and can say, I know and I can prove that this is purple flaming space monkeys number 7. I'm the breeder. This is the individual plant that started it all, and I won the cannabis cup with it, and here it is. So now give us that, and now we know that variety identity code is specifically that variety. And it can be used to provide a means for making a legitimate, provable claim around a variety name and designation. And in plants, you don't generally patent plants. There's different kinds of plant variety protection. And it doesn't necessarily have to be isogenic. There is a certain amount of variability, a certain genetic space, which means there's a little variability in chemotype and a little variability in phenotypic parameters. And we're more familiar with this in things like dogs. 2 Dalmatians look the same. You look at it, you know it's a Dalmatian, even though they're not clones. Well, it's the same with cannabis. And this ability to show identity or very close relatedness has roles in things like IP protection. Something else we've done with it, more out of interest, is something along of what I call a market basket study. And this is a question of what's being sold. Is what's being sold from a producer the same between different lots? Is it even all the same things in a single 1-gram bottle of dried flower sold under one name of a particular thing? And just a quick summary here. What's being sold? A lot of different things that are conflicting names. Is it the same between lots? Sometimes yes, and sometimes no. More often than not, it's yes, but there are exceptions. And is it all the same thing in a 1-gram dried flower bottle? Well, we have seen examples of up to 4 different, completely different things put in one bottle with one name. We've seen that a couple of times. Three varieties one time and 4 varieties another, and both of those out of a single LP, incidentally. Well, I know what you're all thinking at this point. VNTR is totally awesome. Is there anything it can't do? Well, I'm glad you asked that, because yes. And we've sort of touched on this, but we maybe didn't think of it in this form. Remember that I've stressed that VNTR has really no capacity to report linked traits because it's a very small number of genetic loci with hypervariability per loci to give you the power. Because it's this very small number of spots in the genome, it's not a suitable method for marker-assisted selection because, by definition, that requires markers all over the place next to your genes of interest. I would say that doesn't mean VNTR is completely not useful in a breeding program. What we can get out of it is what we call a heterozygosity score. This is a measure of how inbred a sample is. If you're a breeder and you're going to do crosses from parental strains that have particular attributes, particular phenotypes that you like, using inbred parental strains is more likely to pass those traits on than to use less inbred parental strains. So if you have two varieties and they both have this trait you want, but one has a high heterozygosity score and one has a low heterozygosity score, it's more inbred, and you're going to try to do a cross, using the low heterozygosity score, the more inbred parent is more likely to give you a higher fraction of progeny that you want. So it has some utility there, and we have had some customers use that data to do that and to look at inbreeding and back crossing progress. Okay, so to sort of recap, the phrase marketing is using here, and I think it's a good one, is you want to know what you're growing. Don't grow the wrong thing. And this is critical to ensure product uniformity, to meet customer expectations, to build brand identity and build loyalty. Right? DNA fingerprinting, I hope I've explained, is a very powerful and it's an objective means to do this, both of which are important aspects of it. Within DNA fingerprinting, VNTR is a really good method because it gives you the ID power without giving away anything you don't really want to be giving away. And the flip side of this is that it's not having linked traits means that you can't really use it for marker-assisted selection. And again, I do want to hammer back, though, that just because two samples are close on a phylogenetic tree, you can't take any one particular trait. These are close. That was really high THC. Therefore, this is also very high THC. It's probably very high THC, but you could be surprised because as soon as there's a difference, you don't know where that difference is. Okay, well, there's some further resources here, and I'm listing them here. You can see them right now. You can see them if you look at this again, but these are all available if you go to the Segra website. One is on the term genetic drift. How does it apply to cloning and micropropagation? The answer is it doesn't really, and it's misused, and I explain why in that article. Rose by any other name would smell as sweet. I talk about cannabis variety designations and the plethora of names applied to single clones and what needs to be done and why we would like to fix that. Something on marker-assisted selection, although I've just told you today that VNTR is not really a good approach for that. MAS will be useful, and there are other approaches to do that in the future. And we also have I believe, a video of my ICR talk from earlier this year on cannabis variety definitions and what would be needed to demonstrate a cannabis variety. Okay, so I think with that, this is where I'm supposed to be handing it back over to the marketing folks, and they can tell you a little bit about how you would send us a reference sample if you're interested in doing that.
Awesome. Thanks so much, John. Fantastic presentation. I really enjoyed going through everything. And now to the audience, remember, in addition to internal QA purposes, we also offer this DNA fingerprinting on customer cultivars as a validation and plant tracking tool. The slide that we're on now shows kind of a blank version of our certificate of genetic identity that we send out for each cultivar, whether it's run as a standalone sample or as part of our regenerate and preserve service to clean up your genetics. So we'll switch over to the Q&A portion for now. But if you're interested in learning more about DNA fingerprinting, feel free to reach out to Carson and me directly. We'd be happy to chat more about this test and how it's being applied in RMP, the Regenerate and Preserve service, and our genetic supply services today.
Awesome. Yeah, I can fire it off with the first questions here. We have a bunch of them rolling in, which is great. The first one, which is an interesting subject. John, how do you see this technology applying to trademarking cultivars?
Yeah, I think it's crucial to it. So if I were looking at that and I was looking for plant variety protection, you need to show uniformity, true to type of what you're claiming, and you need to show uniqueness. And this is an indisputable way to do both of those. And also to, if you, for instance, sell a plant on, maybe like under a royalty thing, it's a way to make sure that this can't be sold out the back door without you being able to detect that when it's out in the environment. So I think it's a crucial tool in doing that.
Awesome. Thank you.
Another one kind of on something you touched on the start. What is a point mutation and how is it problematic in cannabis cultivation?
So a point mutation is one class of mutation. It's generally a single nucleotide transition. Where they're damaging would be, let me give you a real obvious example. You're growing a cannabis and it's a high THC variety. And if you get a point mutation right on the ATG start codon for the synthase gene for that, something that changed the coding of that first critical amino acid that triggers the machinery to build a protein to do that job. The protein's not made, and suddenly you don't make that enzyme, and yes, there should be 2 copies, but now you're only going to have 1 copy of that gene. So single point mutations can shut genes on and off. Point mutations in a regulatory gene will grossly change or can grossly change overall expression levels of a whole row of other genes, so they can have very wide-ranging effects on phenotype. Having said that, the cannabis genome is, as I said, about 900 megabytes is haploid and about 1.8, 1.9 gigabytes is diploid. And, a single point mutation most of the time is probably not going to do something. You're worried about the ones that do hit these critical bits.
And so essentially it's just a small change in the genome and the idea is that, if it happens in the point where you take a clone and then you grow that clone up into a bigger plant, then it becomes a bigger, proportion of the total plant. Right.
So that's a little bit different. That's more of a founder effect. And that's no reason to think that's a single point mutation. That could be an inversion, a deletion, an insertion, retrotranspose on things. So point mutation is a specific type of mutation. What you're talking about is more founder effect and what happens when you get a mutation at a spot in the plant. So that's a little bit different.
Thanks for differentiating. Carson, you want to jump in with another?
Yeah. On the note of mutations, and a question came here asking about somoclonal variation. So I was hoping you could elaborate a little bit more on how we utilize this tool to monitor for somoclonal variation and ensure what we send out isn't mutated in any way.
Right. So what we do is we routinely monitor plants that are in cultivation. So they get tested right when they come in. We want to capture them right away. And then at regular intervals, we take a statistically relevant sample of that cultivar and we test them. And it's kind of boring on my end because I'll get 40 samples and most of the time 40 out of 40 is exactly the same genotype. And you sit there and you score these things, but that's what you want to see. If we detect, if mutations start occurring and over time, eventually a small percentage will start to get these, we start to detect those and we run some mathematical tests and we can, if this goes above a couple of percent, then we do is we go back, we take 100% verified true to type stuff and we reinitiate off that and then you get rid of the old variety. And it depends in different, or this happens in all organisms. Generally about every 2 years in principle, you want to reinitiate from 100% verified material to ensure that you stay, 95%, 99% plus isogenic.
Awesome. So basically, we spot a mutation, we're getting rid of that, and then just multiplying from the isogenic material that we receive.
Correct. If as soon as we get something, it's called out. That's the idea, or at least I report it over, and I'm assuming somebody over there has a big axe or something. And
That's it. And now you briefly touched on physically marking and tracking the samples. Now I know you're pretty good at it by now, but how long does it take when you receive a sample to actually fully bring it through the VNTR process?
The routine workflow, assuming there isn't anything else in the way. So if I'm just sitting here idle and a sample comes in, it takes about 5 days total. Because it has to go through a sessioning into our database system. We do some checks. There's actually, I won't go into all of it, but we have some error check mechanisms in the sample kit to make sure if somebody's taking a bunch of samples that they didn't give us sample data card A and sample B, because that would screw everything up. And we check that everything, the serial numbers on the tubes and the cards match, and then we enter it in the database, and then it goes through a DNA extraction protocol, which there's an overnight step, and then there's a several hour step the next day, and then we do the actual VNTR process, and then it gets scored, and then it gets analyzed. Occasionally, 1%, 2% of samples, we have to go back and repeat something. We get, there's inhibitors, and so we have to dilute the sample. All told, generally we can do a turnaround's about 5 working days. We generally say 5 working days per group of 16 and an additional working day for each 16 in a batch, but it obviously depends on total workload and total flux in the lab at any given moment.
Okay, that makes sense. Thanks.
On kind of a related note, how many, related to throughput, how many tests have we processed to date?
Around 2,700 with the current version of the assay. And so before that, we had some earlier developmental versions of the assay. So probably another 4, so 3,000-ish total.
And that's a mix of customer samples and internal kind of validation, QA, things like that. Right.
Correct. A lot of it's internal surveillance, these sort of things where, I get 40 of the same thing. But yeah, that's total number of tests that have been run.
Awesome.
And then how scalable is it then?
How many resources can you give me? One of the things we are looking to do, and this isn't really a place to talk about it, but we are looking to do some things to increase throughput through automating some of the scoring. Right now, this is still something that's literally scored by eye, these eyes right here. And I'm very good at doing that, but I'm also kind of slow compared to a computer. And to make this something that's computer scorable involves some R&D and some changes. And as time allows, we're developing that, and it looks very promising. So between some things to automate some of those steps, it's scalable, limited only by your willingness to throw money at the hardware. We have, realistically, we can do hundreds of samples a week, so 800, 1,000 a month with what we have here. But give me money, I could make it 10 times that. I just need more space and more equipment.
And kind of on a related note to that, what other services does our kind of lab division offer? Do you guys run any other tests that might be kind of applicable or relevant here?
Not externally. We do some pathogen testing. We do that internally where it's warranted. But right now, none of that's done externally. We have dabbled in whole genome sequencing. And we've actually published a little bit on that, and there's certainly some very interesting things there. We've looked at some targeted panels of just genes on the cannabinoid synthetic pathway to see what we could get out of that, and it's really interesting. But at the moment, we don't really have the time or resources to be chasing up on that, so that's kind of on hold at the moment.
Definitely, and then kind of a follow-up question there was: has Segra published anything on this topic? But it sounds like we have, and there's
it's all been at this point. It's been posters or presentations at conferences. We haven't published anything in a peer-reviewed journal. We haven't had the time. I'm kept pretty busy just doing day-to-day stuff, so we haven't done that. The general techniques are well understood in the industry and published. There's hundreds of papers on cannabis VNTR typing already.
Fantastic.
This one's a little bit outside the cannabis realm, but so does this technology pass over to mushrooms, and how would it be useful there?
So the technique we're using here is actually the method used for human clinical and forensic identification, if that helps give you an idea. The answer is VNTR identification can be done on essentially any species if there are suitable markers. The test we have is quite specific for cannabis. And by that, I mean, if we put a hops sample, hops is very closely related to cannabis, we put a hops sample in here, it doesn't work. We tried. It would have been nice to get what's known as a genetic outgroup, and most of the markers do not exist in hops, and they simply don't amplify, and we don't get data. The overall method would be applicable to fungi and mushrooms, but you would have to identify and develop those specific markers. And in general, this is used for identifying individuals within a single variety. Within the fungus in particular, most work is done on differentiating one subspecies from another, which is a sort of a higher level of differentiation. And that's usually done by sequencing, I think, the 28S ITS internal transcript spacer 2 region, or something called barcode or live BCOL. So yes, but not directly.
Okay, awesome. Thanks.
Jumping back to one of your examples from earlier in the presentation, one of the questions is about the frequency of mutations just from clonal propagation. I know you mentioned that kind of some examples of it. Do you have any estimates or kind of guesses on the frequency there?
I wish I could give you an answer on that. You'd have to go back to our particular client there. 1 out of 55 showed a very clear single marker. Out of 24 markers, 1 obviously varied. I don't know how many cycles of cutting that had been. What I can say is in tissue culture, it's about 2 years, but I think time is probably part of the equation, because it's things like background radiation are where you get these mutations, and chemical things like cytosine deamination, which isn't detected. But it's also its number of passages. And so I think it's hard to say. Year to 2 year to exceed 2 to 3% is a rough guess, but there's a lot of variables there.
And conditions probably affect that. How much kind of background radiation?
Well, background radiation is one. Other things, like I mentioned, cytosine deamination, that occurs normally, and there's cellular repair processes for that. That occurs thousands of times per cell per day, and almost all of them are fixed, but the ones that aren't, if it happens to be in the wrong place, so to speak, and and that occurs, regardless of background radiation. So there's a lot of different sources of mutation which are an ongoing threat to cells.
Fantastic.
And as for sending in samples, can they be sent from outside of Canada?
So we have a very detailed set of instructions about this. You can send us samples from outside of Canada, but if they are crossing international borders, what you have to do is do DNA extraction on your end. And the little page that marketing people can give you describes the 3 different ways you can send us stuff. So externally, you need to actually do a DNA extract. We tell you roughly what it needs. It doesn't have to be a really concentrated or really even a very clean one. One of the advantages of the VNTR method is that it works very well on dilute and dirty samples. It's extremely reliable. So international, you need to do DNA extraction. If you can't do that, I'm sure you can find a local university or biotech company, which for a couple of dollars will do it for you.
Perfect. Thanks.
Awesome.
And we have done that. We've done that for a couple of U.S. customers. They've sent us DNA. It's actually easier for me because then I don't have to do the DNA extraction step. But does put the onus on them of making sure they properly label tube A, sample A. If they get that wrong, I would never know the difference, right?
Yeah, exactly.
Kind of combining a couple questions about proprietary genetic data into one. Can you kind of recap how certain DNA tests might expose some of that information? If a customer is to send a cultivar in, and then how the VNTR kind of avoids, or doesn't capture any of that information.
So that's actually a very long, convoluted answer. Unfortunately, if you want to understand why, but I'll try to explain it this way. The method, VNTR uses a very small number of points in the genome. So 10 or 12, depending on the version. Right now, 12 points times 2, because there's 2 copies each one. So 24 specific spots in the genome. But each of those spots can have a very large amount of diversity. So it's 24 times this very large number. Each of those can have 10, 12, 14 different versions there. And that's what gives us this very high probability of identity. But you're only looking at this very small number of places in the genome. So most of the genes that are of interest to anybody are nowhere physically near that. And during recombination, when genes reassort between chromosomes, they're a long ways away, and so they're no longer linked. So they're not what's called a linked trait. So we're only looking at a small number of spots in the genome, and they're nowhere near any genes of interest. Things like SNP, on the other hand, use a massive number of spots across the genome. 2,000, 3,000 are kind of reasonable numbers there. And each of those spots has very low diversity. It has maybe like 2 choices. And that's how they multiply number of spots by number of varieties. And that's how they get up into the same sort of total number of genotypes they can detect. But now, because you have these multiple tags scattered all over the genome, the likelihood of one of those tags being very close to a particular gene of interest, serving as a marker, as we would say, for that gene, and co-associating with that gene as it reassociates during breeding, means that it's now linked. And so if you use a method like GBS or SNP, somebody with enough data can mine that and say, you have an extremely high probability of having the following traits based on these markers, whereas there's really no way to get that with VNTR.
Awesome. And then just kind of jumping back for a second to the frequency of mutations, we've had a couple more questions about kind of the rate and frequency in tissue culture. And just to kind of recap on what you said, you said probably about every 2 years is when you kind of start seeing that risk and when you need to reinitiate and double check.
So, I'm not the tissue culture expert here, but I know Dr. Sabayat, our guys, that has told us that for other varieties, it's routine. Somewhere between 12 and 24 months is kind of for pretty much anything in tissue culture. For good practice, you want to reinitiate. And, we have done this surveillance where I get all these repeat samples out of stuff. And samples that have been in TC for around 2 years is where I'll start to see once in a while, oh, you know what? 1%, 2% of this stuff has usually it's one detectable change out of 24. You don't really see more, but that's enough, like I say, to be a difference. And so that tells us that the rate in cannabis in TC appears to be about the same. And as long as we reinitiate every year to 2 years and we monitor for it, we can be confident that what's going out is the right thing.
Awesome.
We've one here, about the specific linked trait. And now, although minimal, what specific linked trait information does a VNTR disclose?
None.
Absolutely none. Awesome. And
none that I'm aware of.
Perfect. And is there any other labs offering this test?
Not that I'm aware of doing it commercially, but I mean, if you go in the literature, this is used for VNTR for cannabis has been around for more than 10 years. Australia, criminal services used it a lot. And in the U.S., different, again, usually state forensic labs used it for legal purposes. Connecticut was, for some reason, a real hotbed of this and a hotbed of research. A couple of big people there. And this thesis was somebody in the State Department of Justice, and it was funded, I think, by the Federal Department of Justice in the U.S. So it's done, but I think, as far as I know, we're the only people who do it to the industry as a useful tool, as opposed as a means of trying to put you in jail.
Fair enough. That's good.
And kind of related, is our test proprietary, or is it?
Well, the way we do it is proprietary. The exact particular set of markers we use, how we analyze the data. I touched briefly on what we call a variety identity code, this 32-character string that we use things on, like the genetic identity certificates. We're the only one who does that. It's an internal tool. So if we had two different clients, they could look at this at each other, and it doesn't tell them anything about it. It just tells them if it's the same thing or not the same thing. So the process is proprietary. The way we analyze the data is proprietary. Overall methods are pretty well understood, but you can only compare results between the exact version of the assay. So if somebody else does a different version of this, we can't really talk to them. What we would do is two different versions of the assay would tend to give you similar phylogenetic trees. They'll both agree on if something is similar. They should both agree on if something is clonal, but I can't look at their data and look at my data, and we can't mix and match those. It's apples and oranges.
Awesome. And, you mentioned the DOJ in the previous question and how VNTR is used in cannabis for more criminal orders, and you've also mentioned this for forensics. What are the other applications for VNTR outside of the use that we use it for cannabis?
The primary use for VNTR is, commercially at least, is in human forensics and human sample tracking. It is, people may have heard of what's called the CODIS database. It's like every crime show wants to mumble, "Oh, he's in the CODIS database." And that, I don't remember exactly what the acronym is, but it's the combined DNA database that uses a particular 16 marker human VNTR thing. It's also used often in cancer pathology labs when you get samples from someone's tumor or possibly a tumor and you're trying to figure out, what it is, and you're looking at it under the microscope. Labs don't like to tell you this, but there's sometimes a problem. You get bits floating around in the processing tank and you're not sure which patient they were from. And now you got to go figure out where it was from. So it's used in the background there to confirm that this chunk of material over there was in fact out of the right person. And it's used for anthropology. So it's really used a lot in human identification. And it's used because it's extremely reliable. It works on very dirty samples. It works on very small amounts of samples. And it's cheap and it's fast. And the data is easy to process.
Awesome. Thank you.
And then kind of related, how do you see this being applied, over the next, let's say, 5 or 10 years in cannabis? Do you see it kind of ramping up? What areas do you see really kind of taking off because of VNTR or improving VNTR?
I don't know if I see it here, but I'll tell you what I would dream of seeing it as. If you go out and get grapes, or you buy a prize winning, a dog from a major breeder, anything like this, variety names have very particular meanings. And it'd be great if cannabis was the same, that if you went to get an OG Kush, you actually knew it was an OG Kush. I pointed to something on the phylogenetic tree where I think we had 19 names on one particular clone. It must grow really well. It must be popular, but we've seen it called 19 different things. It's the same thing. VNTR, because of its identity power and its non-disclosure of linked traits, I think would be a very good method for industries in this space to agree on as to choose a method, choose VNTR as a method and choose a particular panel and analysis approach to that, to have a shared way to identify type specimens as basis of varieties. And just like Dalmatians aren't clones. Within cannabis, you might have multiple things that are not exactly clonal that you still want to call OG Kush. And that, in my opinion, would be based on three things. It'd be based on the DNA relatedness by VNTR being very close within there's a certain allowable amount of variation. Again, they're not clones, right? So there's a certain amount of genetic variation allowable. And within that, I think you would also probably want to say specifically THC and CBD levels and the ratio of those levels. And then I think probably the third attribute would be overall terpene and cannabinoid profile to an experiential perspective, and that's a clumsy way of saying it, but there is a researcher who specializes in this, and I think you'd want to say that a variety has to match the type specimen plus or minus a little tiny bit of variation around all three of those, and I think the VNTR would be kind of the underpinning of that. And so now if you did this, you could have like a international agreement that this is an OG Kush and that's a Girl Scout cookie. We've seen, I think we have 7 different things that have been called Girl Scout cookies. I don't know which one's the right one, but I'll tell you, they're not all Girl Scout cookies. So that's, I think, where ideally it could go in this industry, and I think would do a lot towards brand protection, variety identity. It would protect breeders' rights, and it would make customers know that they're getting reliably the right thing that they want every time.
Fantastic. And, I guess kind of just to probably do one last question that a couple of people have asked about, and stepping back and looking at where we are now in the industry, can you share a little bit of the results from that supply chain analysis that you mentioned where we took a bunch of market samples and tested them? You kind of alluded to it there, but it sounds like there's a pretty good amount of variation among reported names out there.
Well, reported names are all over the place. Yeah. Interestingly enough, if you pick a less common variety name, we tend to see those all tend to be very genetically very close. And I think it's because, it's not a real high value sample. It's not really widely spread. So there's both little reason for intentional mislabeling, which may be one source of error. And there's also, because it's less widely spread, there's less chance for accidental mislabeling because that occurs, once every X many samples. And if there's a lot less samples, it doesn't happen as often. So, but if you pick the really popular name, sour diesel, for example, is more than 2% of all cannabis for sale that we did an analysis a couple of years ago. You pick these really common names, and grower 1 versus grower 2 versus grower 3, we may find that a couple of them have something that is very close to each other. It doesn't necessarily mean it's the right thing, but at least they agree on the name. And then you'll have somebody else that's out there. And then we have these examples, like I say, of one particular clone, which we've seen multiple names. And there are a couple of examples of that one is really specifically, there are several LPs in Canada selling it under wildly different names. The lot to lot variation, generally not too bad. I can think of one particular case where one LP, 2 different lots were not the same material, but most of the time when we've looked at that, they are the same material, or at least very, very close, the kind of thing where I'd say, these are so genetically close that I could believe, these are 2 very closely related mothers, and I'll let it pass. I would accept they're probably the same thing. And that thing where within one bottle, 1 gram sample is it multiple things. We've seen that twice. Those are both from a single LP. Again, I'm not going to say names here, but it suggests that that's their production practices, and they probably know very well that they're doing that.
Awesome.
Yeah, very interesting. Well, yeah, it looks like we're running up on time, but thank you, John, for letting us and through us, the audience, pick your brain through the Q&A period and giving us a great presentation. For everybody in the audience here, if you want more information or if there's anything that we didn't cover, we will be circulating a recording of this, and you can always reach out to us through the info@segra.com. We'll get back to you or confer with Dr. John Brunstein and get the correct answer over to you. So thanks, everybody, and appreciate your time.
Well, it's been my pleasure. Thanks, everyone.
Thanks, guys.